DNA, the double helix, stores genetic information. But storing information is only half of the genetic battle. You also have to be able to take that information and make molecules, make the chemicals of life. For that critical task, all organisms use RNA to manufacture proteins from the genetic information carried by DNA. In Lecture 50, I want to tell you about a second type of nucleic acid called RNA. We're going to see that three different kinds of RNA, messenger RNA, transfer RNA, and ribosomal RNA, work together to assemble proteins. And those are the molecular workhorses of all cells. We're going to learn about the function of genes, which carry instructions for manufacturing proteins. And we're also going to examine the surprising genetic world of the virus. Within a span of half a century, we have learned that every living organism, from slime mold to sequoias to us, shares the exact same language, the same genetic language of heredity, recorded in the form of the molecule DNA. That DNA breakthrough unified genetics by providing a conceptually simple chemical basis for all life. It showed you how we could store information, how we could duplicate that information in the form of a molecule. So we see a logical progression now from cellular to molecular genetics. The molecular structure of DNA, that molecule deduced by James Watson and Francis Crick in 1952, holds the key to its behavior. Recall that DNA has a ladder-like structure. It has 4 different kinds of rungs made up of complementary base pairs, A and T or C and G. That double helix structure has vertical sides to a ladder-like structure. The vertical sides are alternating sugar, phosphate, sugar, phosphate. They make the strong verticals of the ladder, and then the rungs are the base pairs. Because there are 4 different letters that make up the base pairs, A, C, G, and T, you have a 4-letter alphabet that can convey information. And because it's a double helix that can be split apart and have 2 new double helices where before there was 1, you can duplicate information with this molecule as well. The scientific community was captivated by the perfection, by the beauty of this simple molecular design. They embraced the Watson and Crick structure immediately upon its publication in 1953. The discovery of DNA structure marked a historic transition point in the biological sciences. Prior to 1953, when a structure was published, there were various studies of genetics at the level of organisms, at the level of chromosomes, at the level of molecules, and they were carried out by a completely different group. You had botanists and zoologists studying the organisms. You had cellular biologists looking at chromosomes. And then you had people trained in organic chemistry looking at the chromosomes and trying to understand that chemistry. These were completely different training, different groups of people. Suddenly you had the DNA breakthrough, and all of a sudden it unified genetics by providing a common conceptual basis, a simple chemical basis for understanding how genetics works. This transformation in genetics, by the way, is very similar to the transformation in plate tectonics in my own field, the earth science, which in so many ways unified previously disparate ways of studying the earth. Here comes the big challenge, though. Whatever the nature of the genetic code, it had to somehow convert that 4-letter alphabet, the A, C, T, and G of DNA, into a correct sequence of amino acids. And there are 20 amino acids to make a protein. Because after all, the proteins are the workhorses of life. So you have a code on the one hand, which is carried in DNA, and you have proteins on the other hand, and you had to get from one to the other. How do you make proteins? Remember that edict that 1 gene equals 1 protein. Well, progress was made by studying mutations, studying how things work when something goes wrong. Some mutations were known to result in the replacement of 1 amino acid, for example, with another one, and I mentioned sickle cell disease in the last lecture. Other clues were revealed in the late 1950s by pioneering genetic research on mutant forms of the simple bacterium E. coli. What you did is document different mutants, different examples of E. coli when things went wrong. And then you looked at where the genetic words varied before and after, and found out what misspellings in the proteins led to those mutations. It turned out from these experiments that it appeared very likely that 3 letters in each DNA corresponded to 1 amino acid in each protein. And so an additional clue was there. How do you take those 3-letter groupings or codons in DNA and link them up to amino acid? That's called the genetic code. It was during a decade of intense and often highly competitive research that biologists discovered to deduce these details, the process by which DNA leads to the synthesis of a protein. Ultimately, the key to understanding the genetic process is this. You have to take molecular information, you have to convert it into an actual protein. And they discovered that the synthesis depends on a third type of molecule, ribonucleic acid, or RNA. RNA is very closely related to DNA, and there are only a few differences. Here's what they are. Each RNA is a single-stranded molecule of nucleotides, not a double helix like DNA, but a single-stranded polymer. Another difference is that the sugar ribose substitutes for deoxyribose in the long backbone of that polymer. And finally, there's a slightly different base. Uracil, or U, substitutes for thymine, or T, another little wrinkle that you have to keep in mind. So with the exception of those 3 little differences, RNA and DNA are very much alike. There are 3 different forms of RNA that participate in protein synthesis. And first is messenger RNA. Messenger RNA copies the base sequence from DNA, and this is how it works. You ever had one of those zippers, for example, in an old sleeping bag or an old jacket, which is broken so that even though the jacket's zipped up or the zipper is zipped up, there's an opening in the middle? And you see the 2 sides of the zipper sort of exposed where they're not supposed to be? Well, that's exactly what a certain kind of enzyme does to DNA. It unzips a central portion of the DNA and exposes one portion, one gene of the DNA. RNA nucleotides then zip in and they form a template, a copy of that one stretch of DNA. They form a messenger RNA, which is a single-stranded molecule which contains all the information you need to make a single gene. The genetic code then relates to a specific group of nucleotides that correspond to specific amino acids in a protein. And this task was deciphered in the early 1960s by making synthetic messenger RNA chains. For example, you make a synthetic RNA added to a protein synthesizing machinery in E. coli and see what protein the E. coli made when you snuck in these extra messenger RNAs. The first breakthrough came when workers at the National Institutes of Health used a synthetic RNA containing exclusively uracil bases. Now, the unique protein produced was made entirely of the single amino acid phenylalanine. And what that told you was that combinations of 3U, the UUU combination on messenger RNA, corresponded to the protein phenylalanine. If you had a long messenger RNA made only of U, you get a long protein made only of phenylalanine. And that was the first step in deciphering the genetic code. Many additional experiments soon revealed the spelling of all the 3-letter words, the 3 base words or codons of the genetic code. The complete genetic vocabulary consists of 64 different words, 64 different codons. That's all possible 3-letter combinations of A, C, G, and T. 4 times 4 times 4 is 64, and that's your complete genetic code. It turns out that 61 of the codons define specific amino acids, while 3 of the codons mark the end of a gene on the strand or the beginning or have some other punctuation sort of necessary ability. There's one codon, ATG, or AUG on the RNA side, that codes for the amino acid methionine, and that also marks the beginning of all genes. So every protein begins with the amino acid methionine. And so we have that marker to begin every gene, ATG. The code is quite redundant. Remember, there are only 20 different amino acids, but there are 61 different codons that specify amino acids. So in many cases, 3 or 4 or more of those codons specify the same amino acid. So there's a certain safety built in. You can have a certain kinds of mutations and still have the correct protein being built. By 1964, it was discovered that this genetic code is common to every living thing on Earth. Every living organism known uses the exact same genetic code. So you see the genetic code defines a one-to-one correspondence between 3 bases on the one hand and an amino acid on the other. The next step was then to deduce the cellular mechanism by which the information of messenger RNA, which is still just a bunch of information, serves as a template for actually making the protein. So how do you physically construct the protein from amino acids? You have a message stretched out on a messenger RNA, and then you need transfer RNA, the second kind of amino acid. Transfer RNA is a fascinating two-ended molecule. On one end, you have 3 exposed bases, the 3 of a codon. The other end of the molecule has an amino acid. So if you can imagine this structure, 3 ends on one side corresponds to a codon. It links up with an amino acid at the other end, so you can string amino acids at one end corresponding to the sequence of bases at the other. There are 61 different kinds of transfer RNA, each one for one of the 61 different codons, and each one corresponding to a specific amino acid. Now, assembly of a protein with 30 amino acids thus requires you to use 30 transfer RNA molecules. It also requires a messenger RNA unit that has 3 times 30 base pairs, or 90 base pairs long. So you start seeing this correspondence, the linking up of transfer RNA which matches codons to amino acids. And finally, you need a third kind of RNA, ribosomal RNA. The ribosomes are structures in every cell which contain this ribosomal RNA along with about 50 different proteins. This makes a chemical machinery for linking together the amino acids. This ribosome looks something like 2 balls, a smaller one perched on top of a larger one, and so there's a groove where the 2 balls meet. The messenger RNA fits exactly into that groove between the 2 balls. So as the long messenger RNA molecule feeds into the ribosome, appropriate transfer RNA molecules fit into grooves on the side of the larger ball, one after another. And the balls sort of rotate, passing the messenger RNA through that groove. And as it does so, one after another, the transfer RNAs fit onto the surface of the ribosome. They link their amino acids together. And as the messenger RNA is pulled through the ribosome, an assembly of the protein occurs at the bottom side of the ribosome. It's an amazing process. What's really remarkable is that every living thing uses the exact same genetic mechanism. Human genes can be inserted into yeast or into E. coli, and the appropriate human protein will be produced by those simple single-celled organisms. In the short span of half a century, molecular biologists have thus deciphered the 4-letter genetic alphabet. They've learned its complete 64-word vocabulary, and they've even learned a few sentences in that vocabulary. So now we're poised to read the book of life. I want to tell you about the Human Genome Project and the concept of the genome. Every human being has what's now estimated to be about 80,000 genes. Each of those genes is a genetic instruction. It carries coded information to manufacture a protein. So we have in every cell of our body the potential information to manufacture 80,000 different kinds of protein. One of the greatest challenges in modern biology is to identify each of those genes and then to understand the structure and the function of the associated protein to each of those genes. This is an epic ongoing task. It's still really in its infancy. We've only been doing this for a few decades. Only about half of the human genes are even recognized at this point. That's why we don't know whether the number is really 80,000 or 100,000 or what. And only a handful of the known genes are fully described. Early in the 21st century, at least part of this great scientific challenge, though, may have been finally met. After many years of effort, scientists will have at last determined the full record of human genetic information, that is, the human genome. The Human Genome Project is funded in part with billions of federal dollars, your tax dollars at work, and it's coordinated from a base at the National Institutes of Health in Bethesda, Maryland. It addresses this massive problem, which is in molecular biology and in information technology as well. Here's what the project wants to do. It has 2 main objectives. The first is called mapping. The second is called sequencing. Okay, the first of these goals then is to provide a detailed map of the human genome. This is the distribution of genes on each of the 23 chromosomes in every human cell. Now you can imagine this is trying to come up with a roadmap, if you will. Think of each chromosome as a long strand with one base pair after another, and along that strand you have segments, sometimes broken, sometimes fragmented, but different segments that correspond to each of the different genes. And so it's like plotting out a long interstate and recording all the towns and villages that are along that interstate, and you do that for each of the 23 chromosomes. That's mapping in the Human Genome Project. Then the second goal is to determine all 3 billion letters of the human genetic message. That's called sequencing. It's a very automated process now, and it's the new technologies that have sped up sequencing that allow the Human Genome Project to be completed way ahead of schedule and under budget, I might add. The first complete human genome is going to be an eclectic collage of sequences from many, many different people. It was originally thought that maybe the first human genome should be James Watson's genome, but then it was decided that really was not such a good idea to make it just one white male. So people from all races, all parts of the world are participating, and different segments of different chromosomes are identified from these different people. That'll be the first complete human genome. But of course, eventually we're going to have tens and then hundreds and then thousands, countless complete human genomes of many people from many different parts of the world. We're going to start getting more and more information because your genome and mine differ slightly. We're probably 99.9% the same, but there's that slight difference, and that'll be fascinating to see what those differences are. Mapping the human genome, the roadmap aspect, relies initially on the identification of distinctive short DNA sequences, typically 200 to 500 base pairs long, and these are like markers. These are things that are similar, if not identical, in everybody's genome. So these markers can be used to sort of find your way around. It's kind of like mile markers on an interstate. This is an ongoing effort that requires combined skills of molecular biologists who manipulate and analyze the DNA themselves, and also information experts, computer people, because you have to collect and collate huge amounts of information, 3 billion base pairs in one genome alone. You have to search that data for recurrent patterns. You have to look for genes, and then the function of the gene is also often related to certain common sequences that are shared among many different genes. If you imagine finding 2 similar genes in a 3 billion base pair genome, it's sort of like trying to find 2 similar sentences in an entire encyclopedia. It's a huge job, but with computers you can do it. So what they do is they establish tens of thousands of these road markers, or sequence tags as they're called. And then they use this reference system, and that's how you identify locations along all the 23 human chromosomes. And this has been done. The first stage, there are about 30,000 sequence tags by mid-1997. As many as 100,000 of these tags are eventually going to be produced. And then it'll be very easy for researchers to locate specific segments of specific chromosomes and continue with the work in a more systematic way. This is part of the Human Genome Project. Now, sequencing, this second part of the Human Genome Project, is really essentially a problem of information gathering and storage. I mean, collecting 3 billion sequences of letters is a huge amount of data, but it certainly can be done with modern computer technology. Thanks in large measure to a huge influx of genome funding, and also because of the tremendous implications, the economic implications of finding new proteins, new genetic material, many of these processes have been automated, and now it's very, very speedy. So it used to be a cost $1 or more per letter to determine the human genome. Now it's less than 50 cents per letter, and that price is going down. The original target date was 2005. Now it looks like it's going to be 2003, maybe even 2002, when the entire project is finished. Okay, after sequencing, there still remains the massive and mammoth process of determining the function of each of these human genes, of each of the proteins. That task is going to be an agenda for biology, certainly for the next century, and maybe well beyond that. There are just so many proteins, and to determine all those structures is going to be incredibly difficult. But that effort is going to be facilitated by a knowledge of the genomes of lots of other organisms. And so that's also part of the Human Genome Project, to determine the genomes of microbes, of yeast, of worms, flies, mice, for example. Many of the genes of these organisms are quite similar to human genes. For this reason, the Human Genome Project also supports all these other efforts. More than 50% of the genes in yeast are the same as genes that occur in the human body, for example. A chimpanzee is more than 98% genetically identical to us. It's amazing the amount of overlap. In 1996, an international consortium completed the first sequencing of the entire 13 million base pair of yeast, and that was the first complete genome. At first glance, it might seem surprising about all the overlap between different organisms and why you'd want to study yeast or study slime mold or some other things. But remember, every organism uses the same genetic code. So you have to have the same ribosomes. You have to have the same way of building proteins in your body. Every organism uses the same metabolism. Every organism uses the same kinds of cell walls, the same kinds of building blocks. So it's really, if you think from a chemical basis, it's not surprising the overlap of genetic material in humans and other simpler life forms. Preliminary gene catalogs are now giving us a lot of hints about the functioning of different proteins. When you see two genes that have very similar sequences of DNA letters, then you say perhaps the proteins also have similar functions. There's now been a survey of about 10,000 human genes in this preliminary way. It suggests that roughly a quarter of the body's known proteins act as enzymes. These help manufacture cell materials. Then there's 15% that seem to take part in metabolism. There's another 10% that form all sorts of critical cell structures, the actual physical parts of cells. There are other proteins essential for cellular defense, for communication, for reproduction, and so forth. But fully a quarter of all the known genes and the proteins for which they code remain unknown. We just don't have any idea what their functions are. So there's a huge amount in terms of biochemical information that we have yet to learn about genes. This human genome is vastly complex, but you can get a sense of the human genome and how it works by looking at a much, much simpler genome, that of a virus. So I want to spend the last part of this lecture talking about viruses. See, a virus is nothing more than a strand of DNA, or in some cases a strand of messenger RNA in what are called retroviruses. And that's surrounded by a coating of protein. That's all it is, a protein coat with some genetic material inside. Now, depending on who you talk to, viruses are either the simplest living things or perhaps the most complex non-living things. And that's really a semantic distinction. But viruses are not composed of cells, and they can't survive independent of a host cell. So that gives us a hint that maybe viruses really shouldn't be counted quite like a living cell in the category of these biological entities. Nevertheless, under the right environment, viruses can reproduce and they actually evolve. Let me tell you about some of the distinct properties of viruses, and this helps define what they are. First of all, viruses always require a host cell to perform virtually all of its functions. Without a cell, the virus can't do anything like reproduce. Viruses are not capable of independent metabolism. They require to get their energy from a cell. Viruses are much, much smaller than the smallest cell, so viruses can easily enter a cell and inject their genetic material into a cell. Viruses possess either DNA or RNA. They have to have some kind of genetic material, and that carries what's called the viral genome. In retroviruses, that's a messenger RNA, and that messenger RNA strand is converted to DNA by a special enzyme called reverse transcriptase, and that'll come very important when we talk about genetic engineering. This is a specific enzyme that takes RNA and makes DNA out of it, reverse transcriptase. So remember that. We'll get back to that when we talk about genetic engineering. Now, when taken into a host cell, the virus disintegrates. The protein coat just dissolves, and it leaves exposed that genetic material. That viral nucleic acid then takes control of the cell's genetic machinery, and it starts making hundreds of new viruses before the cell dies. Viruses work essentially by fooling the host cell into thinking that it's food. A receptor sees the virus protein coat and says, aha, a nice protein, I want that inside for food. And then, oops, turns out that that protein is attached to a complete virus. The receptor opens the door to the virus's nucleic acid. The cell polymerase then replicates the viral DNA over and over again and makes hundreds of copies of the viral DNA. By the same time, the cell's machinery starts making copies of all the proteins, the protein coats that make up the viruses. And so essentially the cell is co-opted making more and more and more copies of the virus until eventually the cell bursts and dies, flooding the surrounding cells with new copies of the virus. Let's look at a typical viral genome to see how the genetic code works. Now, if you're listening to this lecture on audio tape, there's a picture of this viral genome in your guides. We're going to look at simian virus 40, or SV40, which gives monkey a kind of 24 hours flu. Its genome consists of 5,243 base pairs in a closed loop of DNA, and that closed loop of DNA is very typical of small cells and also of viruses. That SV40 genome contains exactly 5 genes. 3 of them form the protein coat, and 2 of those genes co-opt the cell's machinery and sort of instruct it to make more copies of the virus. The DNA loop has a starting signal to indicate where to start reading. It sort of looks like ta-ta. It's a combination of A's and T's. And then the SV40 genome has a number of features that are also very typical of how genes are distributed on chromosomes. And they're really quite surprising. First, different genes are read in different directions around the loop, clockwise or counterclockwise, if you will. A second trait, one of the genes is split into 2 segments, in this case separated by about 500 base pairs. So you have to start reading that gene and then skip over 500 base pairs and continue reading it. That's very typical, something called an intron, an extra piece of DNA that really doesn't have anything to do. Every organism has this sort of feature. In addition, you'll see that there's substantial segments of several of the proteins that overlap each other. That is, the genetic information to make protein 1, 2, and 3 may overlap in large segments each other. So that means that there are large segments of amino acids that are identical in 3 different proteins. There are numerous viral diseases that affect humans, and they've been the subject of intensive research. That's one of the reasons why viral genomes are so well known. But unlike the DNA in our cells, viral DNA has no correction mechanism. You see, in every cell in your body, DNA is constantly being damaged, but the cell knows how to correct that. In a virus, there's no mechanism to correct damage. So if the DNA is damaged in a virus, it just stays damaged. Therefore, viruses mutate extremely quickly, and viral diseases are constantly changing from year to year and year. There are more than 100 different strains, for example, of rhinovirus, which causes the common cold. Immunity for any one strain, therefore, doesn't prove effective necessarily for other strains, so year after year you may always get colds. You don't develop immunity because new types of colds are commonly coming into vogue, if you will. Similarly, every year there appear new strains of the influenza virus. That's why physicians try to anticipate the mutations of the flu virus and give you a flu shot which has the type of virus that's most likely thought to be coming into prominence in that particular flu season, but sometimes they're wrong. Then there's HIV. That's the retrovirus that causes AIDS. It attacks the human immune system so that the body actually loses its ability to immunize itself against other infections. Ultimately, the cause of death in AIDS is not so much the virus itself, HIV, as it is the other diseases that exploit the body's weaknesses. Many viruses can affect cells and remain in a dormant state, perhaps by inserting its genetic material directly into a cell's genome. So you have episodic diseases such as herpes, which may reappear from time to time. Sometimes years go in between recurrences. They can be triggered by any number of factors, including stress and sunburn. I get these sort of mouth sores every so often when I eat chocolate. It seems like chocolate triggers the virus, but otherwise it stays dormant. So this is something we all have to kind of deal with and learn what factors may trigger viral diseases in your own case. There's so many questions about viruses that remain unanswered. We don't know how they originated, for example. We don't know what their role has been in evolution, and it may be significant because viruses may be able to insert genetic material into organisms that didn't exist there before. And as we'll see in a couple of lectures, we can now potentially use viruses to alter our own genomes. In summary, while DNA carries the code of life, there are 3 kinds of RNA that use that code to make proteins. We have messenger RNA that copies the gene from DNA and brings it to the place where proteins are made. We have transfer RNA that matches up 3 letters of the genetic code to 1 amino acid, a key step in making proteins. And finally, we have RNA, ribosomal RNA, that assembles the proteins. In the next lecture, we're going to see how progress in understanding the role of genes has been made on many different fronts.